Hi everybody, welcome back. Today we're going to be looking at an introduction to DNA for forensic science. Now let me say up front, this is not meant to be a comprehensive video on DNA function or structure. If you're looking for that, go elsewhere. All we want here today is a basic introduction of some of the most important details of DNA for somebody who's looking to gain a basic understanding of how DNA analysis is done for forensic science. With that out of the way, let's begin. So, first question is, how is DNA used in forensic science? What's the general principle? The general principle is that we compare DNA that's left behind at the crime scene to DNA from victims or suspects. So, for example, perhaps we find the skin cells of some other person underneath the fingernails of a dead victim. Well, it wouldn't be unreasonable to conclude that perhaps those skin cells got there because the victim clawed at the face of the perpetrator while the murder was being committed. And if that's true, then the murderer's DNA is underneath the victim's fingernails. So if we can compare the DNA under the fingernails to someone we suspect as the murderer, and we find a match, well then that's a strong piece of evidence linking that suspect to the crime. We can also compare the DNA from the crime scene to a DNA database. So folks who commit certain crimes will have their DNA profiles added to a database, and even if we don't have a particular suspect, we can still analyze the crime scene DNA and check it against a database, and many crimes have been solved this way. So let's emphasize, what we're looking for here is a match. We want a match either between the crime scene DNA and a hit in the database, or the crime scene DNA and someone we suspect as being involved in the murder. Now, what if we found DNA at the crime scene, but we don't have any idea who to suspect? We find some person with no personal connection to any others in the middle of the woods. There's no evidence of what happened here, but we have some other person's DNA present. Where do we start to look? And then imagine there's no hits in the database either. How would you know whose DNA that is? Well, the short answer is you wouldn't. Unless you have someone to compare the crime scene DNA to, you're kind of out of luck. Now, maybe later on, that person will kill again and they'll get caught and their DNA will be entered into the database and you can look it up. But unless you have somebody to compare the crime scene DNA to, that's it. It's a dead end. So just like with fingerprints, we needed to compare the fingerprints found at the crime scene to the fingerprints of a suspect. Same thing here. We need to compare the DNA found at the crime scene to someone we suspect is involved in the crime. So we can use DNA evidence as part of a criminal investigation. We can also use DNA evidence later on after a conviction as part of an appeals process. And I want to introduce you to something called the Innocence Project. Let's take a look. So here we are on the website of the Innocence Project. The Innocence Project is an organization that that their mission is to overturn what they see as faulty convictions. In other words, cases where a person's been convicted of murder or rape or other serious crimes and they've gone to jail for a very long time, but those people have been falsely accused and convicted without sufficient evidence. And what the Innocence Project does is people will send requests to the Innocence Project saying, I'm innocent of this crime, I've been serving for 10 years, please help me. And what the Innocence Project will do is they'll go in and they'll talk to the people, they'll look at their case records, they'll find out, was the lawyer representing this person actually doing a good job? And then relevant to our work today, they'll ask, was DNA analysis done? And if DNA analysis has not been done, and it looks like it would be relevant to the crime, say a knife was picked up from the crime scene and it's got the blood of the perpetrator on it, if that was never tested for DNA, the Innocence Project will pay for DNA testing to compare the perpetrator's blood on the knife to the suspect who's been serving a sentence for 10 years in prison. And if they find that, in fact, there's no match, they'll bring that evidence back before the court and they'll try to get the conviction overturned and the person freed. So we can take a look at some of the cases that they've worked on here, and by all means, Google or go to this URL here at the top, go to the Innocence Project, read about these cases. It's stirring stuff, and I think anybody who, if you're watching this, you have some interest in the justice system, it's inspiring to see when the justice system gets it right, when it frees people who haven't committed crime, when it puts people behind bars who deserve to be there. But we ought to keep in mind that we don't always get it right. The justice system has flaws, and it's good that there are people who are working to exonerate those who have been falsely convicted. And one of the key pieces in their work at the Innocence Project is DNA evidence. So it can be used to get a conviction. It can also be used to exonerate a person who's been falsely accused. All right, so that was a little bit of a sidebar, but I think it's important to think about these things. Let's get back to the DNA. Okay, so we saw in the Innocence Project that if the person who was convicted of the crime, if their DNA actually doesn't match the DNA that was found at the crime scene that we're pretty sure was the murderer's, the rapist, or whatever, then that's pretty good evidence that they really shouldn't be in prison for this crime, and they may be exonerated. But we ought to ask the question, what if the crime scene DNA does match the suspect's DNA? What sort of conclusions can we draw? So imagine that a DNA analyst has come to you, and the DNA analyst has said, I've got a perfect match here. I've run the sample of the suspect, I've run the sample of the crime scene, they match up perfectly. What are the possibilities here? Well, one possibility is that the suspect actually wasn't there, and the DNA analyst made a mistake. That's possible, isn't it? People do make mistakes. This is good technology, but nobody's perfect. People do make mistakes. It's possible the suspect wasn't there, but somehow their DNA ended up there. Maybe they were framed. Maybe somehow their tissue got there, even though they were never there. It's possible. It's possible the suspect was there, but had nothing to do with the crime. In other words, perhaps this person walked through the scene, got a paper cut, left some droplets of blood at the scene of the crime, and then left. And then later on, someone was murdered in that room. If that's the case, you'll find their DNA there, even though they had nothing to do with the crime. It's possible the suspect was there, but had a lesser role in the crime than you suspected. Maybe they were the getaway driver, not the murderer. And then, of course, it's possible that the suspect was there and had exactly the role in the crime that you suspect. So, it's important as scientists, forensic scientists are a type of scientist, as scientists, we must always consider all of the possibilities. We cannot simply say, well, the analyst said that there's a match, that means that this person is guilty of murder. No, we have to consider all the possibilities. And it may very well be that the most likely possibility is that in fact the person committed the crime. But we always must be open-minded to all the possibilities and give each one a fair shake. All right, moving on. Our next question is, what is the function of DNA? And you might be wondering, well, why do we care for the sake of forensic DNA analysis? And the reason is, without some knowledge of the function of DNA, and in particular the structure of DNA, it's going to be very, very difficult for you to understand how forensic DNA analysis is carried out. And I'm going to try to keep this to just the minimum of information that's really needed for this project we're undertaking. But without further ado, let's get into it, the function of DNA. So the function of DNA. DNA is the stuff that encodes the information that tells dog cells how to make a dog, that tells cat cells how to make a cat, that tells banana cells how to make a banana tree, or apple cells how to make an apple tree. It's, in other words, the differences between the DNA in different organisms is what accounts for and produces the differences among the different species. It's also what accounts for the similarities in closely related individuals. Here's the Hemsworth brothers, and you can see there's a close resemblance between them? Well, no coincidence, they're siblings. They share a disproportionate amount of DNA together compared to random strangers in the population. So it's no surprise that they share the same jaw structure, or the same eyes, or the same general type of hair, and so forth. So let's dig in a little deeper. DNA basics. DNA stands for deoxyribonucleic acid. It's present in every cell in your body. And in every cell, the number of DNA molecules is 46. What do we call DNA molecules? We call them chromosomes. Where is the DNA stored? It's stored in the nucleus of each cell. And what it does is it stores the information needed to operate each cell in your body. It gives the instructions to the liver cells, how do you be a liver cell? It gives the instruction to the brain cells, how do you be a brain cell, and so on. Now, a little bit on the structure. Each DNA molecule or chromosome is made of two very, 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 very long strands, which are intertwined in a sort of double helix shape. Those strands are themselves made of smaller subunits which fit together to make the very long strands, and those subunits are called nucleotides. It takes millions of nucleotides to make a single molecule of DNA like you would find in one of your cells. There are 4 types of nucleotides: adenine, thymine, guanine, and cytosine, or if we want to be brief, just A, T, G, and C. The way that DNA stores information is in the sequence of these nucleotides, in much the same way that a book you'd pull off the shelf stores information in the sequence of letters. The difference between War and Peace and Harry Potter isn't that the symbols used are different, it's just that the number and the arrangement of those symbols, the number and the arrangement of the letters is different. And hence you've got either a story about a boy wizard or a tome of a piece of Russian literature. All right, moving forward, let's take a look at some images that might help to clarify some of these pieces we've just learned about. So here is an image of onion cells under a microscope that have been treated with a dye that stains DNA purple. And you can see these purple blotches. These are the nuclei, the nucleuses, if you like, inside the onion cells. It's lighting up purple because that's where the DNA is. We can zoom in a little further and we catch some cells in the process of division. And most of the time you can't see this, but when cells are dividing, the chromosomes become visible. And you can see them in this image as these little sort of wiggly, worm-like shapes in some of these cells. Well, we can actually take the chromosomes out of the cells when they're in this form and examine them individually. That's called making a karyotype. Let's take a look at a karyotype now. Here you have it. Here are your 46 chromosomes. Go ahead and count if you like. There are 46 of them here. What I'd like to do now is show you a chart that organizes them by type. So it's actually the case that of your 46 chromosomes, each one has a partner, or has a pair. In other words, in your 46 chromosomes, there's actually 23 types of chromosome and 2 copies of each type. So if you look at the diagram here, we've got in the upper left chromosome 1. There's 2 copies of chromosome 1. Immediately to the right of chromosome 1, the 2 copies of chromosome 1, there's chromosome 2. And you can see there's 2 copies of chromosome 2. Look midway down the page, there's chromosome 15. You've got 2 copies of chromosome 15. So you've inherited one of each chromosome from your mother and one of each chromosome from your father. This becomes really important later on when we look at short tandem repeat analysis to understand that in fact you have 2 copies of each chromosome, and each copy contains generally the same information. What I mean by that is that your chromosome number 1 may contain information on how to make the protein hemoglobin in your blood, may have genes that specify your height, or the type of muscle tissue that you generate. Well, your other copy of chromosome 1 also contains information specifying how to make hemoglobin, or your height, or the muscle tissue that you produce. The versions of that information may be a little different. In other words, one version of chromosome 1 may have the hemoglobin that works really well, and the other copy of chromosome 1 may have a hemoglobin that doesn't work so great. But the general idea is that each chromosome in a pair has the same information as the other chromosome in the pair. So chromosome 1, whether it came from your mom or your dad, contains the same sorts of information. Information about height, information about hemoglobin, and so on. Those are just made-up examples, but I hope it starts to clarify the point. When we look at these chromosomes and we zoom in on them, we actually see that these large structures are made of smaller threads, and we can keep unwinding these threads. And what we eventually end up with as we unravel and unravel and unravel and unravel these threads is the familiar double helix shape. This is the so-called twisted ladder, the 2 strands of DNA which are wrapped around each other, the classic double helix. Let's take a closer look at that classic double helix. Here's your double helix. Often what we like to do to further understand the structure of DNA is actually untwist this helix. And when we do that, we get what we'll call a DNA ladder. Now, this is not how DNA exists in your cells. It's just a way of presenting it to make it a little easier to see the structure. And so what you can see here now are that the uprights of the ladder, the vertical pieces, are what we call the sugar phosphate backbone. But the rungs of the ladder, if this were a ladder, the places you put your feet, those are the really key pieces. Those are the nucleotides. Let's take a look at those in even greater detail. Here now we've taken the DNA ladder and we've added a little more molecular detail. These gray circles represent phosphate groups. These blue pentagons represent deoxyribose sugar, and all the labeled shapes in orange, yellow, green, and purple represent the nitrogenous bases. And so these nucleotides all together represent adenine, thymine, guanine, and cytosine. Really, really important vocabulary moving forward. A pair of these we call a base pair. Okay, so a pair of these is called a base pair. And you have a grand total of 3 billion base pairs per cell. So what you're looking at on the screen right now is 4 base pairs. You've got 3 billion per cell. That's a lot of base pairs. So, a little more about the sequence of the bases. The information in your genome is encoded in the sequence of these bases. In other words, do we see AAA, TGC, CG, CCA, or do we see GCC, TAA, AGC, TT, ACC? All right, what's the order of the letters? If we take all of the DNA in one of your cells and put it all together, we call all that DNA, all the information it encodes, all the molecule, we call that your genome. And your genome contains about 3 billion base pairs of nucleotides. As a metaphor, your genome is like a library with a theme, and the theme is how to make a human, specifically, how to make you. Each molecule of DNA, or remember the term, chromosome, contains the information to determine many different traits and contains an average of about 100 million base pairs. So every molecule, 100 million base pairs. These are big molecules. Here's your metaphor. Each chromosome is kind of like a book in the library, and the topic is a variety of different things you'll need for making a human, specifically for making you. You may wish to keep in mind at this point that the chromosomes are not organized by trait. In other words, it's not like this chromosome tells you how to make blood cells, and this chromosome tells you how to make liver cells, and that chromosome tells you how to make brain cells. The traits are, near as I can tell, rather randomly distributed across the chromosomes, with the exception of the sex chromosomes, which we can get to later. Each trait is encoded by what we call a gene. This is a length of DNA that's on average about 17,000 base pairs in length. As the metaphor, each gene is like a chapter whose topic is one thing you'll need for making a human, specifically you. A few more notes. The genome is really big, and it's sometimes difficult to understand quite how big. This becomes important in forensic science. It gives us a sense of how big the task is of actually comparing one person's genome to another person's genome. So if each gene were really a chapter, and it had 17,000 characters, it would have about 2,800 words. That's about half the size of your average Harry Potter chapter. It'd be about 12 pages. An above average reader would take about 9 minutes to read that. So the genes are, there's a good chunk of information there. If each chromosome were really a book, it would have about 17 million words and would span 67,000 pages. That's about 20 times the length of the entire Harry Potter series, from the very first book all the way to the last one. If you were a fast reader, it would take you 120 days, 8 hours a day, to read the whole thing. That's pretty big. Now, if the genome were really a library, it would have about 500 million words and would span 2 million pages. The book would be 200 meters tall, or 640 feet tall, and would weigh 9,000 kilograms, or about 20,000 pounds. This is a lot of information. If you were to read it 8 hours per day, it would take you 9.5 years to get through the whole thing. What's the takeaway here? Your genome is really big and contains a lot of information. Let's take a look at 0.002% of the human genome, shall we? So here we are. I've loaded up the first 60,000 letters of the human genome. This is the first part of chromosome 1, if you're curious. So here you can see all those A's, T's, G's, and C's, and we'll scroll down through them, and you can see that this is really quite a large thing. And this whole thing is just 0.002% of your whole genome. And we're about halfway through it right now. Nearly there. There we are at the end. So what you've just seen scrolling past is 0.002% of your genome. 0.002% of all the 3 billion base pairs that make up your genetic identity. Pretty amazing. So you've just seen a sneak peek of just a snippet, just the tiniest fraction of your genome. You've seen it's really, really big. Well, how are we going to actually make comparisons from person to person? It turns out that of those 3 billion base pairs, depending on how you count, 99.9% of them are identical from person to person. In other words, if we took your DNA and my DNA and we compared them, 99% of those letters would be in the same place and they'd be the same letter. We look at, base pair number 38 million in chromosome 1. It's a T in me, it's almost certainly a T in you, and so on. So we're almost identical. But if 99% of our base pairs are the same, that means 0.1% are different and unique. In fact, no two people have exactly the same DNA, with the exception of identical twins. Forensic DNA analysis uses this 0.1% uniqueness to distinguish one person from another. So, we're nearing the end of today's video, but I want to give you a little sneak peek, just a taste of what's coming next, how DNA analysis really works. So the basic idea is this. we're going to compare the base sequences, that's the A's, the T's, the G's, and the C's, that pattern from the crime scene DNA with the same pattern from the suspect's DNA. And we're going to look, are they the same or not? If the base sequence is exactly the same in the crime scene DNA and the suspect's DNA, then that means it's their DNA. So what's the process? What's the general process? Okay, so first DNA is extracted from collected samples. You get your samples from underneath the fingernails, you take a blood test of the suspect, and so on. Then we process the DNA samples, and the goal there is to make those very few differences among the samples visible, so that we can actually distinguish two different people's DNA. Once we've done that, we can compare the DNA samples from the crime scene to the DNA samples from the suspect, and we can see if there's a match. So next time, our goal is to answer this question. How do we do this? How do we process DNA samples to make those very, very small differences visible? And then how do we compare it to see whether our suspect was really there at the crime scene? Let's summarize. DNA analysis compares DNA collected from the crime scene with DNA collected from suspects. Each person carries an astonishing 3 billion base pairs of DNA in every cell. These 3 billion base pairs of a person's genome are distributed across 46 molecules, which we call chromosomes. The 3 billion base pairs are made up of the nucleotides A, T, G, and C. That's adenine, thymine, guanine, and cytosine. The sequence of the nucleotides encodes the information of the organism. In other words, it's the order and the identity of the letters A, T, G, and C, or the molecules that they represent, that determines what information is stored in the DNA. It's those very few differences in each person's genome, the sequence of the letters, that forms the basis for forensic DNA analysis. And then again, to summarize the process, forensic DNA analysis consists of DNA extraction, processing of samples, and then finally comparison of samples. Next time, we'll start taking a closer look at how this work is actually done. I hope this was helpful. We'll see you next time. Bye for now.